Trophy, it is nothing but here a single gene which shows multiple phenotypic expression. Pedigree analysis is nothing but the practice of analyzing inheritance pattern or trait in several generations of family. Myotonic dystrophy is a condition which affects the muscles. So it is a disease which affects the muscle. Hello everyone, a warm welcome to today's session on chapter 5, that is Principles of Inheritance and Variation. I am Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session, we had studied about something called as polygenic inheritance. So polygenic inheritance is nothing but wherein 3 different genes or more than 2 genes, they determine or control the character or trait of the progeny or the individual. But in today's session, we shall deal with one very important aspect which is called as pleiotropy. So there is a slight difference between polygenic inheritance and pleiotropy. So pleiotropy, it is nothing but here a single gene which shows multiple phenotypic expression. So here one single gene, it controls the different traits or characters in an individual. Coming again, what is polygenic inheritance? In polygenic inheritance, multiple genes control one single character or a trait, wherein we took the example of the human skin color. But when it comes to pleiotropy, one single gene controls different characters or traits. So I can explain this by taking the example of a disease called as phenylketonuria in humans. So phenylketonuria, it is a birth defect usually seen in young children wherein the amino acid, that is phenylalanine, starts accumulating in the body. So there is one amino acid called as phenylalanine. So this phenylalanine, which is a amino acid. So it is a amino acid. So this phenylalanine is needed for the body. So the phenylalanine is the one which produces the one more amino acid called as tyrosine. So there is one enzyme called as phenylalanine hydroxylase, that is phenylalanine hydroxylase. So this phenylalanine hydroxylase is the one which converts the phenylalanine to tyrosine. So tyrosine, why is it needed for the body? Because tyrosine actually maintains the skin color of an individual, and tyrosine is not produced, the individual will have a skin disorder called as eczema. And also for the proper functioning of the brain, for the neural control, all that this tyrosine is very, very important. But what happens in the case of phenylketonuria disease is that there is accumulation of the amino acid phenylalanine. So now you can ask me, ma'am, if there is accumulation of the phenylalanine, it is good, right? Tyrosine, a large amount of tyrosine will be produced. So that is where the fault lies actually. What happens there is, anything too much is always bad. How much it has to be produced, so much only it has to be produced for the proper functioning of the body. It's just like too much calcium in a body is also dangerous, right? Just like that. Here, phenylalanine, when it starts to accumulate, what happens is, it will result in phenyl deficiency. It will result in phenylalanine deficiency. Therefore, what happens? It cannot convert, the phenylalanine cannot get converted to tyrosine, which is needed for the various functioning in a body. So therefore, what happens? The child will develop skin diseases such as eczema, the proper functioning of the brain will not occur, the skin color will change, all that will happen. So that is what I said, what is pleiotropy? Here one gene, that particular gene that is responsible for the production of the enzyme phenylalanine hydrolase, which helps in the conversion of phenylalanine into tyrosine, it will get altered. So when it gets altered, what happens? Phenylalanine will not be produced, too much amount of phenylalanine will be produced, therefore it will hinder the process of the development of one more amino acid, tyrosine. Therefore different diseases can be seen in the human body. Therefore what happens here? One single gene is controlling different traits or different characters. That is nothing but pleiotropy. Next, moving on to mutation. So mutation is one of the leading factors that is responsible for causing a wide range of diseases. You know that nowadays the food we eat, it is not good. It is filled with pesticides. It is filled with harmful fats and oils and harmful sugar, right? So what happens here is, as the year goes by, when we start consuming such products, or even the pollution, the exposure to radiations, all that will lead to mutation in the DNA. The best example for this mutation disease is the cancer. Cancer is nothing but cancer occurs in the body because of mutation. So where does this mutation occurs? It occurs in the DNA. So we have the DNA. So if I write this DNA is a helical structure, but here I'm just drawing it in a linear strand like this for easy understanding. So you know that DNA has various nucleotides, A, G, T, A, C, A, G. It has various nucleotides, adenine, guanine, thymine, cytosine. It has various nucleotides, right? Here what happens when mutation occurs is, it will change in the genotype. It will change the genotype. So whatever genes are there, it will alter the genes on the DNA. Therefore, it will bring about a change in the phenotype or the morphological character of the individual as well. So mutation is nothing but a sudden heritable change in the DNA sequence which brings about a change in the genotype and the phenotype of an organism. So this mutation actually leads to variation in the DNA. It brings about changes or variation in the DNA. It is always specific. So it's like in a DNA, a nucleotide should be arranged in a specific manner. If that specificity is gone or if it is altered, then what happens? It will result in the variation in the DNA. Therefore, it will cause various diseases. So mutation will result in variation in the DNA. What kind of variation? Either there will be a loss or gain in the nucleotide. So for example, maybe this nucleotide may be lost. It will be lost. So when it is lost, you may have only A, C, A, G combination. Sometimes in between, some other nucleotides may get added. Say for example, A, C, G, A, T may get added here. So either the nucleotides may get deleted, or a part of the DNA may get deleted, or a part of the DNA may get inserted. So when it inserted, what happens here? A, G, T, A. In between, again A, C, G, A, T, C, A, G. Like this, it gets inserted. So either deletion may take place or insertion of a new DNA strand or new set of genes or new set of nucleotides may occur. So it leads to variation in DNA, wherein either loss or deletion of DNA may occur, or gain or insertion or duplication of the segment of DNA may occur. So this will result in alteration in the chromosome. Why alteration in the chromosome? Because the chromosome has nothing but the genetic material, which is the DNA. So where is DNA present? It is present in the chromosome itself. So it may cause alteration in the chromosome. So that these will result in abnormalities. So alteration in the chromosome will result in a lot of chromosomal disorders. So what are the different chromosomal disorders? We shall see in the next coming session. So there are various causes of mutation. So the first one is, or different types of mutation can occur. First one is the frame shift mutation. So frame shift, one particular frame or a small portion of the DNA is shifting. Say for example, as I told you, you have ATGCA on the DNA strand, you have like this. So maybe this particular portion will shift somewhere else. So maybe here TAGC, like this, you'll have a stretch of DNA strand. So it is shifting here. So what will happen in the frame shift? AT, this T, it will continue TA, somewhere in middle here, this will come and shift here. So GCA, GC. So like this, shifting a portion of the DNA, as such it will shift to some other position on the DNA strand. That is nothing but frame shift mutation. So this shifting will also bring about mutation or alteration in the DNA. So next is point mutation. In point mutation, what happens here is only one nucleotide will either get deleted or one more nucleotide will get added in that particular position. So maybe this may get deleted, or instead of T, G may come here. So here point mutation, only one nucleotide. Understand the difference. Frame mutation or frame shift mutation, one stretch of DNA strand will as such move to another position on the DNA strand. But in point mutation, only one nucleotide is considered, either A, G, C or T. Only one nucleotide is considered. That one single nucleotide may get deleted or may get inserted. That is what happens in point mutation. Next is chromosomal abnormalities. So in chromosomal abnormalities, what happens here is, so you know that we humans, we have 46 chromosomes, that is 23 pairs of chromosomes. So 23 pairs, and these pairs are nothing but we have chromosomes like this in pairs. So two pairs. Pair means it is 2, right? So sometimes what happens during chromosomal abnormality is 1/3 pair chromosome may come here. So this is an abnormality, or one chromosome may get deleted. So that is nothing but chromosomal abnormality. Next is some mutagens. What are mutagens? Those chemicals or those compounds that bring about mutation in the DNA are called as mutagens. So mutagens, there are two types of mutagens, physical mutagens and chemical mutagens. So physical mutagens means you have UV radiation, then X-rays, then alpha rays, beta rays, gamma rays. So we have alpha rays, then we have beta rays, and we have gamma rays. So these are the different types of radiation. So these UV radiation, alpha rays, beta rays, gamma rays, then X-rays, etc. Subjecting yourself to too much X-ray radiation will also lead to mutation. That is why soon after your X-ray is done, you will be asked to come out of the path of the radiation. So physical mutagens, these are caused by UV radiations, alpha, beta, gamma radiations, then X-rays and all these things. Next, moving on to chemical mutagens. So chemical mutagens, usually mustard gas, then phenol, formalin. So formalin, you all are, as a biology student, all of you knows about formalin. So formalin is nothing but those chemical compound that is used in preserving specimens, in like plant specimens, animal specimens, we preserve in glass bottle, right? Inside that glass bottle, they'll put a liquid. That liquid is nothing but formalin. So these formalin, it is actually volatile. It evaporates. So when it evaporates, no, if we take too much of the formalin fume, it is not good for the body. It will cause mutation. That is, it will bring about alteration in the DNA, therefore causing chromosomal abnormality. So these are the different mutagens that are present. So next, moving to pedigree analysis. So what is this pedigree analysis? So pedigree analysis is nothing but to analyze. You might have heard about proofreading. So you'll write an essay. So you want to know what mistakes you have done in the essay. So that particular essay, what you have written, no, that sheet you'll take it to a proofreader and you'll ask him, check the mistakes and let me know. So that proofreader, what he'll do, no, he'll use certain symbols which depicts the mistakes. So just like that, in pedigree analysis also, certain symbols are used. So before moving on to the discussion about the symbols, we shall see what pedigree analysis is. So pedigree analysis is nothing but the practice of analyzing inheritance pattern or trait in several generations of family. Like So pedigree analysis means what? Pedigree, you know about pedigree. It is nothing but your great-grandfather, great-great-grandfather. Then comes your great-grandfather, then your grandfather, your father, you. Like this, the family tree. It is nothing but the pedigree. So this pedigree. So say for example, in a family, you have acquired a disease. Say for example, sickle cell anemia. So sickle cell anemia, it is a hereditary disease, means you have taken it from your parent. So your parent might have taken it from his parent, the parent from his parent, like that. So from where it has started, how the progeny is inherited these diseases, that is nothing but they'll put a chart using these symbols. So that is nothing but pedigree analysis. So the practice of analyzing inheritance pattern of traits in several generations of family is called as pedigree analysis. So here, to go in for pedigree analysis, there is a chart that is represented using a chart is done using various symbols. So the chart, I'll explain it to you later. Before that, we will study the symbols. So the representation or chart showing the family history is called as the family tree. So we have the family tree. So that chart we'll study later. So that is nothing but pedigree. That particular family tree is called as pedigree. So in human genetics, pedigree study is very, very important because it will make us, by putting that pedigree chart, we shall understand exactly from whom did we inherit that particular disease or that particular trait, whether it was from the father or whether it was from the mother. If it was from the father, how did the father get the disease, from whom, like that we will understand very clearly. So that is why pedigree analysis is very, very important. So in human genetics, pedigree study, it provides a strong tool and this particular pedigree tool, it can be used to understand from where the inheritance or from where the progeny has inherited or got a particular trait or a abnormality or a disease. So when I explain using the chart, it will be even more clear for you. So here, first we shall study the different pedigree symbols and then we will move on to the chart. So to represent a male, they have used a box square which is blue in color. So blue color means everything is normal. The male doesn't have any disease. It is a normal male. So to represent a normal female without any disease, they have used a circle. And to represent a sex unspecified, because person is neither a male nor a female, that is in between sex, then it is represented like this. It is represented like this, neither square nor circle but a diamond shape. And the affected individuals, it is represented in red because they are affected, they are carrying a disease. That is why they are diseased, they have a disease. That is why it is in red. So this is the diseased male, diseased female, then unspecified. So unspecified sex, disease, unspecified sex. So now this means mating. Can you see here? There is a connectivity drawn here. So mating between the male, connectivity female. So mating between the male and the female. Okay. So mating between relatives. Again here, you should know the mating between relatives, because it is usually said that if you marry within in the relatives, it will lead to some chromosomal disorders in the child or in the progeny. So to depict that, they have male and female, both are relatives. They are from the same family. They are relatives, very close relatives, like maybe the mother's brother. The female will marry the mother's brother. So like that, very close relatives. So it is represented like this too. They have a very strong connection. It is represented by like this. And parents above and children below are represented like this. So these are the male parent, female parent, and the female progeny and the male progeny. It is represented like this. And parents with male child affected with the disease, it is represented like this. So this is the normal male parent, normal female parent, and the progeny, it is diseased. So it is represented like this. So here what has happened, I'll tell you how you can understand this. So I told you mating means there is a line drawn like this, right? So here normal male and the normal female has mated, and this has resulted in the development of two progenies, normal female, normal male. It is represented like this. And if it is, sometimes if the progeny is diseased, then normal male and normal female, they have mated, but the progeny is diseased. Why? Because it is represented in red color. And if the offspring, that is sex unspecified, 5 means the 5 of the offsprings which are sex unspecified, they are unaffected offspring. So that is this one. This is how the symbols that are used in pedigree analysis. So now we shall see how a chart is being created. So for this, let us take the example of genetic disorders. What is genetic disorders? Any alteration occurring in the DNA or in the chromosome will lead to genetic disorders. So it is of mainly two types, that is two types, Mendelian disorders and chromosomal disorders. So Mendelian disorders and chromosomal disorders we shall see in detail in the next coming class. So I'll take the example of two disorders here. One is myotonic dystrophy, and the other one is sickle cell anemia. So we'll put a chart for, that is pedigree analysis chart for myotonic dystrophy and sickle cell anemia. So I hope you know now what is that round symbol that is red in color here. This is female, right? Why female? See here, female is represented in round and affected female is represented in red color round. It means that the female is affected female. So affected female with normal male. Now what has happened there? A line is there in between. Can you see? There is a line drawn. Means they have mated. Mating has occurred. So when mating has occurred, progenies can be many types. So since the female is having a disease, that is myotonic dystrophy. So what is myotonic dystrophy? Myotonic dystrophy is a condition which affects the muscles. So it is a disease which affects the muscle. So myotonic dystrophy, the female is having myotonic dystrophy and the male is normal. Both have mated. So the progenies, they can either, if it is a female child, she can be healthy without the disease. If the male child can have a disease, or the male child may not have the disease. So these are the possibilities of the progenies, okay? And the female may not have the disease, and again here female child may develop the disease and the male child may not develop the disease. So this is the pedigree chart that we have got till here now. Now male, the normal male and the female, they have mated. So how will the resulting progeny be? If it is a girl child, they are free of disease, and if it is a boy child, they are also free of disease. Why? Because here there is no disease in both of these, right? Now if a diseased female again mates with a male, then what happens? The resulting pedigree, either two male will be disease free. Why? Because it is dominant here, and if it is a female, she will have a disease. So that is how the pedigree chart is done. So again the same way in sickle cell anemia. Sickle cell anemia, it is a condition which doesn't depend on the parents. Like say for example, the parents will have the affected chromosome carrying the gene responsible for sickle cell anemia. But the parents will not have the sickle cell anemia. When the child is born, then that trait or character will be carried to the child. Therefore, the child will develop sickle cell anemia. So here, normal male, this is normal male, normal female, both do not have the disease. They don't have the disease, but they have the chromosome that is carrying the gene that is responsible for the disease. So here what happens when they mate, the resulting progeny, either if it is a male child, he may be free of the disease. There are possibilities. It is not necessary that he should get the disease. It depends on what kind of chromosome he receives. It is depending on that. Whether he has received the abnormal chromosome, then he will develop the disease. If he has not received the abnormal chromosome, then he will not develop the disease. Here, the child may be diseased. Now again, no disease in the male and the female child may be diseased, no disease in the male child. Now, if a mating occurs between these two, that is the normal male and the female, again, here what happens, in spite of them not having the disease, the progeny will be diseased. Why? Because they will have the chromosome, but the disease will not get expressed in them. The gene will remain silent. It will not express the particular disease. That is why, though they are having the chromosome, abnormal chromosome that is responsible for the sickle cell anemia, and they do not have any symptoms of sickle cell anemia, they do not have the disease, but in spite of them having the chromosome, it will be transferred to the progeny. So, the male may have the disease, the female may not have the disease. This is how the chart is done using symbols. So, this is about the pedigree analysis. So, in this particular chapter, we had dealt with especially pleiotropy is nothing but wherein one particular gene will control the different characters, wherein I explained taking the example of the disease phenylketonuria. Then we've studied about mutation, different types of mutations, and also we studied about the pedigree analysis and the symbols that are used in pedigree analysis, and how a pedigree analysis chart is created by using the symbol, so that we can find out exactly from where the disease started. So, actually here the disease started from the parent itself, from the first set of parent itself the disease started, and it got carried to the next coming generations. So, that is what is analyzed using the pedigree analysis. So, I hope you have understood the session very well. So, in the next coming session, we shall deal with the disorders, that is the genetic disorders, wherein we shall study about the Mendelian disorders and we shall study about the chromosomal disorders. So, this is a very interesting session. So, I hope this session I have made it clear. So, you have understood the session very well. We shall meet in the next coming session. Thank you.